Hey everyone, and welcome to the deep dive. Are you ready to kind of jump into a whole new world of learning? We've got something super special for you in this series.
We're going to explore some pretty cool topics together, but not in the way you might expect.
Yeah, we're not just skimming the surface here. This is a 5-season deep dive.
5 whole seasons dedicated to helping you truly understand these complex topics.
We designed this for learners like you, people who are really hungry for knowledge and love that aha moment when everything clicks.
To make sure those clicks happen, we're using a method called spiral learning.
It's kind of like climbing a spiral staircase.
Yeah.
You know, each season takes us higher, revisiting these core concepts with a fresh perspective and going even deeper each time.
So we'll build a solid foundation and then layer on more knowledge and nuance with each season.
Exactly. By the end, you'll have this rock-solid understanding of these fascinating topics.
But that's not all. We're incorporating cutting-edge AI, too, with Google's NotebookLM.
Oh, yeah. So that means you can actually interact with this podcast.
Yeah. Imagine you're listening and a question pops into your head.
Yeah.
With NotebookLM, you can pause, ask your question, and explore further right then and there.
It's like having a personal research assistant right there with you.
We're also going to have quizzes throughout the series.
This is not about testing you or anything. It's about helping you see what's sticking and where you might want to spend a little more time.
Think of this whole experience as a choose-your-own-adventure, but for your brain.
We'll be covering everything from the intricacies of drug development and something called CGMP to the world of receptors and signaling pathways.
Now, I know those might sound a little intimidating right now, but trust me, they're going to become your new favorite topics.
Oh, yeah, they are. And these aren't just abstract concepts. They're incredibly relevant to everyone's lives.
Think about it. We all take medications at some point.
Right, whether it's a simple over-the-counter pain reliever or something prescribed by a doctor.
How much do we really understand about how those medications are made?
That's where CGMP comes in, current good manufacturing practices.
It's basically like a super detailed rule book for anyone making pharmaceuticals.
So everything is carefully controlled and documented.
It's about ensuring that medications are safe, effective, and consistently produced to the highest standard.
CGMP helps guarantee the quality of the medications we rely on. And that trust is especially important when we talk about therapies, using things like cannabinoids and psychedelics.
You know, those substances making headlines for their potential to treat things like depression, anxiety, and addiction.
These therapies are developed through a long and complex process.
It's like navigating this maze of regulations and scientific hurdles.
But first, we need to understand how those substances actually work in our bodies, which leads us to those cannabinoids and psychedelics.
These substances interact with complex systems in our bodies.
One in particular is the endocannabinoid system, or ECS for short.
This system plays a role in a ton of important functions like pain perception, mood regulation, and even how our immune systems work.
Psychedelics, on the other hand, can have a profound effect on how we perceive and experience the world around us.
Research suggests they could be really effective tools for managing things like depression, anxiety, and even addiction.
But to make sure these new therapies are safe and they actually work, they have to go through rigorous testing, and that's where the FDA comes in.
The FDA, or Food and Drug Administration, makes sure any new drug, including those using cannabinoids or psychedelics, is put through its paces.
They evaluate everything based on tons of data from clinical trials before they're made available.
Those clinical trials are carefully designed studies with human participants, and they're absolutely essential to understanding how a potential drug works and what its potential risks might be.
There are different phases to clinical trials, each with specific goals.
Early phases focus on whether the drug is safe for people to take and what the best dose might be.
Then as we move to later phases, those studies involve many more participants and aim to confirm how well the treatment works and catch any potential side effects.
But conducting these trials isn't enough. We also have to make sense of the data.
Right. So things like statistical analysis and understanding how research works become really important.
Drug development isn't just about finding a substance that seems to work.
It's about understanding how that substance interacts with the body at a molecular level.
And that's where receptors and signaling pathways come in.
Imagine receptors as those tiny antennas on the surface of our cells, constantly searching for specific molecules.
Think of those molecules like hormones or neurotransmitters. They're like messages being sent between cells.
When a molecule finds its matching receptor, it triggers a chain reaction inside the cell.
That chain reaction is what we call a signaling pathway, and it can cause all sorts of changes within the cell.
It might lead to the production of new proteins or even changes in how genes are expressed.
It's like sending a message from one part of the body to another.
Different receptors trigger different pathways, leading to a whole range of effects throughout the body.
Some receptors help regulate our mood, while others are involved in controlling things like heart rate or muscle contractions.
Understanding these pathways is crucial for developing new drugs that can target specific receptors to either enhance or block their effects.
And this knowledge has led to some of the most important medications we have today, from painkillers to antidepressants to drugs that help manage blood pressure.
But the world of receptors and signaling pathways is vast and complex.
We're still unraveling all the intricacies involved.
That's why research in this area is so vital.
It helps us learn more about how our bodies work and opens up new avenues for developing therapies that can improve everyone's health.
And as we delve deeper into this world in the coming seasons, we'll encounter all sorts of fascinating receptors.
Receptors involved in our senses, receptors that regulate our immune systems, and even receptors that play a role in the development of cancer.
It's going to be an amazing adventure.
It really is. Welcome back to the Deep Dive Learners.
It's great to have you back for part 2 of our journey. Now remember how we talked about CGMP in the last part? Those current good manufacturing practices are super important for making sure our medications are top-notch.
We touched on how companies use some really cool techniques to spot even the tiniest impurities. But quality isn't just about purity.
Right, it's also about potency. You want to know that medicine is going to pack the right punch. So how do we make sure that a drug is actually strong enough to work?
That's where potency assays come in. Think of them as a measuring stick for how much active ingredient is actually in a drug.
Oh, so it's like checking the caffeine levels in your coffee. You want that perfect energy boost, not too weak and not too jittery.
That's a great way to think about it. Too little active ingredient, and the medicine might not work, but too much could be dangerous. Potency assays make sure each dose has just the right amount to be safe and effective.
That makes sense. But I'm picturing scientists in lab coats with like bubbling beakers. How do they actually figure out the potency of a drug?
Well, there are a lot of different methods, but one of the most common is called high-performance liquid chromatography, or HPLC for short.
HPLC.
Yeah.
Okay, honestly, that sounds a bit intimidating.
It might sound complicated, but it's actually pretty clever. Imagine you have a bag of colorful candies, all mixed up. HPLC is like sorting those candies by their color and size.
So it separates out the different parts of a drug.
Exactly. Scientists inject the sample into this special machine, and it separates all the components based on their chemical properties. Then a detector measures how much of each component is present.
Oh, so it's like each part of the drug is running a race, and HPLC tells us who the winner is and how much of it crossed the finish line.
I like that analogy. By looking at those results, scientists can figure out exactly how much of the active ingredient is in that drug sample.
That's so cool. It's like having this high-tech detective making sure our medications are up to par.
And this is super important not just for traditional pharmaceuticals, but also for those exciting new therapies using cannabinoids and psychedelics.
Right, as those therapies become more mainstream, it's crucial they meet those same high standards as any other medication.
And that means using those rigorous quality control measures, including those potency assays we talked about.
Okay, let's say we are working on a new treatment for chronic pain that uses cannabinoids. What kinds of challenges might we face when it comes to figuring out the potency?
That's a great question. Remember how we talked about botanical drugs being like an orchestra? Each compound is like an instrument playing its part.
Right, and just like an orchestra needs each instrument to be perfectly tuned to create that beautiful, harmonious sound, a botanical drug needs the right balance of compounds to really be effective.
Exactly. Unlike traditional pharmaceuticals, botanical drugs often have a complex mixture of compounds, not just one single active ingredient. So we have to figure out how those different compounds interact. Some might work together, others might actually counteract each other.
So potency testing for botanical drug is much more complicated. It's not just about measuring one single ingredient. It's about analyzing the whole composition.
You got it. And for that, scientists use something called mass spectrometry, or MS.
Okay, mass spectrometry. I've heard that term, but I'm not quite sure what it is.
Think of it as a molecular weighing scale. It measures the mass of individual molecules with incredible accuracy. It's like being able to identify each musician in an orchestra just based on the weight of their instrument.
So instead of just knowing we have a violin, we know exactly which violin it is and how heavy it is.
That's a fantastic way to think about it. By analyzing the weight of those molecules, scientists can identify and quantify each compound.
So they get a detailed roster of the entire orchestra with all their instruments and their weights.
Exactly. And that information is crucial for making sure each batch of that botanical drug is consistent and meets those potency requirements.
It's amazing how scientists have developed these techniques to ensure quality.
It speaks to how dedicated people are to protecting public health and making sure these medications are safe and effective. Now, let's revisit polypharmacy that we touched on before.
Right, the idea that multiple compounds in a botanical drug might work together to create that synergistic effect.
Yeah.
So instead of one compound doing all the heavy lifting, they're teaming up.
Exactly. And this teamwork can be incredibly beneficial, potentially leading to enhanced effects and even fewer side effects.
Can you give us a specific example of how this polypharmacy thing actually plays out in a botanical drug?
Let's stick with cannabis. Remember those terpenes that give different strains their unique aromas like pine or citrus?
Yeah, I love that some strains smell like a walk in the woods.
Well, research is suggesting that those terpenes might actually influence how those cannabinoids interact with the body's endocannabinoid system.
So the terpenes are like the stage managers guiding those cannabinoids to exactly where they need to be.
Perfect analogy. For example, some terpenes might enhance the pain-relieving effects of THC, while others might help CBD better manage anxiety.
That's fascinating. So by understanding these interactions, we can develop even more effective cannabis-based therapies.
Absolutely. Polypharmacy offers a world of possibilities for harnessing the full potential of botanical drugs.
It really goes to show that nature has a lot to teach us.
Now let's dive deeper into receptors and signaling pathways. Remember those receptors like tiny antennas on the surface of our cells? Well, one important group is called tyrosine kinase receptors, or RTKs.
RTKs, okay. What makes these receptors so special? What's their role?
Think of them as the communication hubs of our cells. They receive signals from the outside world and transmit them to the inside, triggering this whole cascade of events.
So they are like the cell's personal assistant, taking messages and making sure everything runs smoothly.
Exactly. And those messages can be about all sorts of things, from telling the cell to grow and divide, to instructing it to change its function, or even to self-destruct.
Wow, it's a lot of responsibility for one little receptor. They sound pretty important.
They are. RTKs are essential for some of the most fundamental processes in our bodies, which makes understanding them crucial for understanding both health and disease.
Can you give us an example of an RTK and how it works its magic?
Let's talk about the epidermal growth factor receptor, or
Yeah.
EGFR.
EGFR rings a bell. I think I've heard of that one in connection with cancer.
You are right on the money. EGFR is one of the most studied RTKs, and it plays a key role in how cells grow and multiply.
So if something goes wrong with EGFR, it could lead to uncontrolled cell growth, like in cancer.
Exactly. In many types of cancer, EGFR is either overactive, meaning there's too much of it, or it's mutated and gets stuck in the on position.
Oh, so it's like the cell's growth accelerator is jammed down and it can't stop speeding up.
That's a great way to put it. This uncontrolled growth is a hallmark of cancer. But the good news is scientists are working on ways to target EGFR and put the brakes on those out-of-control growth signals.
That's amazing. It's like finding a way to cut the engine on that runaway car.
There are already several drugs available that specifically target EGFR, and they have been effective in treating certain types of cancer like lung and colorectal cancer.
It's incredible how understanding these tiny molecular mechanisms can lead to those life-saving treatments.
Absolutely. Now let's turn our attention to another fascinating group of receptors called G-protein coupled receptors, or GPCRs for short.
GPCRs. Okay,
Yeah.
that term definitely sounds familiar, but I need a refresher on what makes these receptors so special.
Well, get ready to be impressed. GPCRs are arguably the most important family of receptors in the human body. They're involved in a mind-bogglingly wide range of functions.
Like vision, smell, mood regulation, even immune responses. Wow, that's quite a resume.
They're the ultimate multitaskers, and they get their name from how they interact with this special type of protein called a G protein.
So it's like a dynamic duo working together to get things done.
Precisely. When a signaling molecule like a hormone or neurotransmitter binds to a GPCR on the cell surface, it sets off this chain of events. The GPCR activates that G protein, which then interacts with other proteins inside the cell.
So it's like a relay race.
Mm-hmm.
The GPCR passes the baton to the G protein, which then sprints off to activate other players in the cell.
That's a great way to visualize it. And depending on the specific GPCR and G protein involved, that chain reaction can lead to a huge variety of responses from the cell.
Can you walk us through a real-life example?
Let's look at beta-adrenergic receptors. These GPCRs are found in your heart, lungs, and other tissues, and they respond to those hormones, adrenaline and noradrenaline.
Ah, adrenaline. That's the fight or flight hormone.
Exactly. When adrenaline binds to those beta-adrenergic receptors in your heart, it triggers a G protein that increases your heart rate and makes your heart pump harder.
So it's like adrenaline is giving your heart this pep talk, telling it to get ready for action.
Exactly. That's how our bodies prepare to deal with those stressful situations. But these beta-adrenergic receptors do even more than that. They're also involved in regulating breathing blood pressure and even metabolism.
Wow, that's a lot for one receptor to handle.
They are incredibly versatile, and that's why they are such important targets for medications. Pharmaceutical companies have developed a wide range of drugs that specifically target these receptors.
Like beta blockers for high blood pressure.
Exactly. And antihistamines for allergies. Even some antidepressants work by interacting with GPCRs.
It's amazing how understanding these microscopic mechanisms can lead to such a diverse array of medications that help manage our health.
It's a true testament to the power of scientific research. Now, I know we've talked about different families of receptors like RTKs and GPCRs, but I've also heard this term receptor subtypes. What exactly are subtypes and why should we care about them?
Yeah, what are those all about?
Receptor subtypes add another layer of complexity to this world. That's like this. Within each family of receptors, you can have multiple subtypes. They all have the same basic design, but with subtle differences.
Okay, so it's like different models of the same car.
Yeah,
They all have the same basic features, but with variations that affect their performance.
I like that. For example, remember those beta-adrenergic receptors? Well, there are actually three main subtypes: beta 1, beta 2, and beta 3.
So 3 different versions of the same basic receptor. Each one has its own unique job.
Precisely. Beta 1 receptors are mostly found in the heart where they control heart rate and how forcefully the heart pumps.
Oh, right, that's the one adrenaline targets to get your heart racing.
You got it. But then you have beta 2 receptors, which are more common in your lungs where they relax the airways and make it easier to breathe.
That's why some asthma medications target those beta 2 receptors to open up the airways.
Exactly. And then there are beta 3 receptors, which are involved in regulating metabolism, especially in fat cells.
So even within a single family of receptors, there's this amazing diversity.
And this diversity allows our bodies to respond to such a wide range of signals with this incredible precision. But it also adds another layer of complexity when it comes to drug development.
Right, because you don't want a drug that targets all the subtypes that could lead to unwanted side effects.
Exactly. The goal is to develop drugs that are highly selective for a particular subtype. Think of it like designing a key that will only fit one specific lock, even though there are other locks that look very similar.
So scientists are like expert locksmiths, crafting those keys to target the exact receptor they want.
It's all about understanding those nuances of those receptor subtypes to develop safe and effective medications.
Now, earlier you mentioned this idea of biased agonism. It sounds pretty technical, but also intriguing. Can you explain what that means and why it's so revolutionary?
I'm glad you asked. Bias agonism challenges our traditional understanding of how drugs interact with those receptors. We used to think of it as this simple on-off switch.
Right, the drug either activates the receptor or blocks it like flipping a light switch on or off.
Exactly. But biased agonism shows us it's not that simple. It's more like a dimmer switch where you can control the brightness of the light.
Oh, so instead of just turning a receptor on or off, a drug can fine-tune its activity.
Exactly. It's the idea that a drug can bind to a receptor and activate it, but it might preferentially trigger some signaling pathways over others.
So it's like having a conductor who can choose which sections of the orchestra to play at any given time, highlighting certain instruments while others are silent.
That's a fantastic analogy, and this selectivity can have a huge impact on the drug's effects.
I'm starting to see why this is so revolutionary.
Hmm.
But can you give us an example?
Let's look at opioid receptors. Think about traditional opioid painkillers like morphine. They activate multiple signaling pathways, which leads to both pain relief and those unwanted side effects like respiratory depression and constipation.
Yeah, those side effects can be really serious.
Absolutely. But what if we could separate the good from the bad? What if we could target only the pathways responsible for pain relief while minimizing those dangerous side effects?
That'd be amazing. Less pain without the risks.
Well, research has shown that it's possible to develop drugs that do just that. We call these biased agonists.
So they're like super selective painkillers, targeting only the pathways we want while leaving the others alone.
You got it. And that's why biased agonism is so exciting. It offers the potential to create drugs that are both safer and more effective than traditional medications.
This is blowing my mind. It seems like we're learning to control even the most complex systems with incredible precision.
It's really remarkable, and it opens up this whole world of possibilities for treating diseases and improving human health. But as we get deeper into these topics, we also have to remember that our bodies are constantly adapting and responding to those signals they receive.
Right. Our cells aren't just sitting there passively, they're actively adjusting to their environment.
Exactly. And one of the ways they do this is through a process called receptor desensitization.
Receptor desensitization. Okay,
Right.
that sounds a bit like our cells are saying, okay, we've had enough of that signal for now time to tune it out.
You got it. It's essentially a way for cells to regulate their sensitivity to signals. If a receptor is constantly being bombarded with a signal, it can become less responsive over time.
That makes sense. Otherwise, our cells would be in a constant state of overdrive.
Exactly. And there are a couple of different ways this desensitization can happen. One common mechanism is receptor phosphorylation.
Okay, phosphorylation. Remind me what that is again.
Remember how we talked about phosphorylation being like flipping a switch on a protein?
Right, it can turn the protein on or off.
Well, when a receptor is activated, it can be phosphorylated by certain enzymes, and that phosphorylation can trigger a whole chain of events that ultimately lead to that receptor being pulled inside the cell.
So it's like the cell is saying, okay, this receptor needs a break. Let's take it off the front lines for a bit.
Exactly. Once that receptor is inside the cell, it can be recycled back to the surface later, or it might even be broken down completely.
It's like those receptors are on a rotation, taking turns being active and then going backstage for a little R&R.
That's a great way to think about it. And this process of desensitization and resensitization helps cells maintain a balanced response to all those signals they're receiving. It prevents them from being overwhelmed and ensures they can keep responding appropriately.
It's incredible how our bodies have these finely tuned mechanisms for regulating everything.
Absolutely. And this constant movement and adjustment is essential for keeping everything running smoothly.
So what happens to those receptors while they're backstage? It sounds like there's a whole other world of activity going on inside the cell.
You're absolutely right. And that brings us to another fascinating concept: receptor trafficking.
Receptor trafficking. Now we're talking. It sounds like a cellular road trip.
Exactly. It's all about the movement of those receptors within the cell. They're not just static entities, they're constantly being synthesized, transported, modified, and even degraded.
Wow, they are busy little molecules.
And this constant movement plays a crucial role in how they function.
Can you give us an example?
Let's look at AMPA receptors. These receptors are found in the brain, and they're essential for learning and memory.
I think I've heard of those. They help strengthen or weaken the connections between neurons, right?
You got it. It's called synaptic plasticity, the ability of those connections or synapses to change over time. AMPA receptors are key players in this process.
So they're like the brain's architects constantly remodeling those connections based on our experiences.
That's a great way to put it. Now here's where receptor trafficking comes in. Research shows that AMPA receptors are constantly being shuttled between different compartments within neurons.
So they're like tiny commuters hopping on and off the cellular train system.
Exactly. When a synapse is activated, those AMPA receptors are inserted into the membrane at the receiving end of that synapse. This makes that connection stronger. It's like adding more lanes to a highway, making it easier for traffic to flow.
But what happens when the synapse isn't being used? Do those receptors just hang out there?
Nope. When that synapse is inactive, those AMPA receptors are removed from the membrane, which weakens that connection. Think of it as those highway lanes being closed down, slowing down the traffic flow.
It's amazing how these tiny molecular movements can affect something as complex as learning and memory.
It really highlights how dynamic our brains are.
We've talked about how those receptors can be shuttled around, but how do they actually get inside the cell in the first place?
That's where endocytosis comes in. Remember how we compared endocytosis to a cell eating or drinking?
Yeah, it's like the cell is taking in little packages from the outside world.
Exactly. And in the case of receptors, endocytosis plays a crucial role in regulating their activity.
So it's like the cell saying, okay, we need to bring this receptor inside for a bit, either for a tune-up or to retire it altogether.
You're right on the money. Once the receptor is inside the cell, it can either be recycled back to the surface or broken down and removed.
It's like a sophisticated sorting center, making sure those receptors are either refurbished or sent to the recycling bin.
It's all about maintaining that delicate balance and making sure the cell is responding appropriately to all those signals it's receiving.
This is all so incredibly complex, but also so fascinating. I'm starting to see how all these pieces fit together. But I'm guessing it gets even more intricate than this, right?
You bet. Now imagine a cell is receiving multiple signals all at the same time. How does it make sense of all that information?
That's a good question. It would be like trying to listen to several conversations at once.
And that's where we get to another amazing phenomenon, crosstalk.
Crosstalk. It sounds like we've entered the world of cellular espionage. Are those signaling pathways spying on each other?
Well, in a way, yes. Crosstalk refers to those interactions between different signaling pathways. They aren't isolated, they can influence and even interfere with each other.
It's like all those different departments in a company trying to coordinate their efforts, sometimes working together, sometimes competing.
That's a great analogy. Think about a cell trying to decide whether to grow, divide, or just hang out and do its thing. It might be receiving signals telling it to do all three.
So those pathways have to talk to each other and figure out what the cell should actually do.
Exactly. That's crosstalk in action.
Okay, so can you give us a concrete example of how this crosstalk plays out?
Let's consider the interplay between the insulin signaling pathway and the inflammatory signaling pathway.
Okay, insulin is involved in regulating blood sugar levels, right?
That's right. And inflammation is a natural process that helps our bodies heal and fight off infections.
Two very important processes, but how are they connected?
Well, research suggests that chronic inflammation can actually disrupt the insulin signaling pathway.
So it's like inflammation is throwing a wrench into the works.
Exactly. This interference can lead to insulin resistance, a condition where cells become less responsive to insulin.
And that means glucose builds up in the blood, which can ultimately lead to type 2 diabetes.
Wow. So something that seems unrelated to blood sugar, like chronic inflammation, can actually have a huge impact.
That's why understanding these interactions is so crucial. We need to look at the bigger picture and how all these different systems in our bodies are connected.
It really highlights how amazingly complex our bodies are.
Absolutely. And this complexity is also reflected in how cells regulate their own activity through something called feedback loops.
Feedback loops. That takes me back to high school science class. Can you remind us what those are and how they work in the context of cells?
Think of feedback loops like a thermostat in your house. When the temperature gets too low, the thermostat kicks on the heater to warm things up. When the temperature gets too high, it shuts the heater off to cool things down.
So it's like a self-regulating system.
Exactly. In a feedback loop, the output of a process can actually influence its own input, either amplifying or dampening that signal. Okay, that makes sense. But how does this work in cells?
Let's look at a system called the hypothalamic pituitary adrenal axis, or HPA axis for short.
HPA axis. Okay, that sounds pretty intense.
It is a complex system, but it's a great example of a feedback loop in action. The HPA axis controls our response to stress.
So it's like our body's internal alarm system.
Exactly. When we're stressed, a part of our brain called the hypothalamus releases a hormone called CRH. CRH then travels to the pituitary gland, which releases another hormone called ACTH. And ACTH triggers the adrenal glands to release cortisol.
Cortisol. Isn't that the stress hormone?
Yes. Cortisol has a number of effects on our bodies, like raising blood sugar and suppressing the immune system. It's like preparing the body for that fight or flight response.
So when we're stressed, our bodies get this surge of cortisol to help us deal with the situation. But what happens when the danger has passed?
That's where the feedback loop comes in. Cortisol can travel back to the hypothalamus and pituitary gland and tell them to stop releasing CRH and ACTH.
Oh, so it's like a built-in shutoff valve. Once those cortisol levels are high enough, the body gets the message to ease up on the stress response.
Exactly. This feedback loop ensures that our stress response is carefully balanced, preventing both too much and too little cortisol production.
That's so cool. It's like our bodies have their own internal checks and balances to make sure everything stays in equilibrium.
Absolutely. And this delicate balance is maintained through a variety of mechanisms, including a fascinating process called signal amplification.
Signal amplification. It sounds like we're turning up the volume on those cellular messages.
You're right on track. Signal amplification is a process where a small signal can trigger a much larger response within the cell.
It's like a domino effect, where one event triggers this cascade of increasingly larger events.
Exactly. In many signaling pathways, each step in the cascade can activate multiple downstream molecules, leading to an exponential increase in the signal's strength.
So it's like one tiny whisper becoming this booming roar.
That's a great way to put it. Remember that cyclic AMP signaling pathway we discussed earlier?
Vaguely. Remind me how it works again.
Well, when a hormone or neurotransmitter binds to its receptor, it activates an enzyme called adenyl cyclase.
Right, and adenyl cyclase makes cyclic AMP.
Exactly. And here's the amazing part. A single molecule of adenyl cyclase can produce thousands of molecules of cyclic AMP.
Wow. That's a massive amplification.
It is. And that cyclic AMP then goes on to activate other enzymes, which in turn activate even more molecules, creating this incredible chain reaction of amplification.
So it's like a chain letter. But instead of spreading gossip, it's spreading cellular signals.
That's a fun way to think about it. And this signal amplification is what allows cells to respond so effectively, even to very small amounts of a signal.
It's like our cells have their own built-in megaphones to make sure those messages are heard loud and clear.
It's another example of how beautifully efficient and elegant our biological systems truly are.
I'm starting to see how all these pieces we've talked about, receptors, signaling pathways, feedback loops, and signal amplification all work together to create this incredibly intricate and dynamic cellular communication network. It's like a grand symphony of molecular events.
And remember, this is just the beginning of our journey. We've laid a solid foundation, and now we're ready to dive even deeper into this fascinating world.
Welcome back to the Deep Dive. It's so great to have you here for our final part of our very first episode.
We've covered a lot of ground, from the basics of receptors and signaling pathways to those quality control measures that ensure our medications are safe and effective.
Yeah, it's amazing to see how all these pieces connect. But I have a feeling we're about to go even deeper into this world of cellular communication.
You are absolutely right. We're going to zoom in on one of the most fundamental processes in our bodies, signal transduction.
Okay, signal transduction. It sounds kind of like a high-tech communication system.
Yeah.
Can you paint a picture of what that looks like inside our cells?
Imagine your cells as bustling cities, constantly receiving information from the outside world and responding accordingly. These messages come in all forms: hormones, neurotransmitters, growth factors. They're all trying to deliver instructions to the city.
So those are like the messages being sent to the city, but how do they get through the city walls?
That's where those receptors we've been talking about come in. They're like the gatekeepers, each one specifically designed to recognize a particular message.
Oh, so it's like having different mailboxes for different types of mail. You wouldn't want your bills ending up in the mailbox for love letters.
That's a great way to think about it. When a message or ligand binds to its matching receptor, it sets off this chain reaction inside the cell, like a carefully choreographed domino effect.
And that chain reaction is what we call signal transduction.
Exactly. It's the process of converting that message from the outside world into a language the cell understands.
So it's like a universal translator, turning the language of hormones and neurotransmitters into actions the cell can take.
I love that analogy. This translation process involves some pretty amazing molecular acrobatics. You have proteins interacting, enzymes being activated, and even genes being switched on or off.
Wow, that sounds incredibly complex. It's like this perfectly synchronized ballet with all these molecules moving in perfect harmony.
That's a beautiful way to describe it. And this intricate dance allows our cells to respond to a constantly changing environment and maintain that delicate balance we call homeostasis.
Now, we've talked about different types of receptors, but are all signal transduction pathways the same, or do they have different styles?
Just like there are different types of dances, there are different types of signal transduction pathways. Each one has its own unique steps and rhythms.
So some pathways are like fast-paced waltzes, while others are more like those slow and graceful tangos.
I love how you're thinking. One of the most well-known pathways is the cyclic AMP pathway, which we've touched on a few times.
Right, that's the one activated by a lot of different hormones and neurotransmitters, and it can lead to changes in how genes are expressed.
Yeah, exactly. Another important one is the MAP kinase pathway. This one is involved in things like cell growth proliferation and even how cells respond to stress.
Oh, right. I've heard that the MAP kinase pathway is often involved in cancer.
You are absolutely right. When this pathway gets dysregulated, it can contribute to uncontrolled cell growth, which is a hallmark of cancer. That's why understanding this pathway is crucial for developing new cancer treatments.
So scientists are trying to find ways to interrupt those signals and stop those cancer cells from growing out of control.
That's the idea. And then there's the JAK-STAT pathway. This one's activated by different types of signaling molecules called cytokines and growth factors.
Okay, and what does the JAK-STAT pathway do?
It plays a key role in the immune response and in how cells differentiate or become specialized for different functions.
Wow, so many different pathways, each with its own unique role to play. It's like a grand symphony orchestra with all these different sections working together to create a harmonious sound.
What a beautiful way to put it. This incredible complexity and interconnectedness are what makes signal transduction such a fascinating and endlessly challenging field of study.
I can imagine. But it's also a field that holds so much promise for understanding human health and disease.
Absolutely. The more we understand about these intricate conversations happening inside our cells, the closer we get to unlocking the secrets of diseases and developing new and innovative therapies.
This deep dive has been mind-blowing. We've explored everything from the building blocks of medications to the molecular dances happening inside our cells, and it's all thanks to those amazing receptors and signaling pathways.
And remember, this is just the beginning. This is a 5-season journey, and with each season, we'll spiral back to these concepts, adding more depth and nuance to our understanding.
I can't wait to see what we uncover.
And remember, learning is a collaborative process. With NotebookLM, you can interact with these episodes, ask your questions, and explore further.
Think of it as your personal learning adventure. We're just here to be your guides. So stay curious, keep those brains buzzing, and we'll see you in our next deep dive.